So now, all of that stuff, primary, secondary, tertiary, that was like outside the EOC, added extra, no extra charge. I'm sure you're excited. Here's some more that's no extra charge. This is for your health. Here's one big reason that we've discovered that sugar is really the bad guy in nutrition and not fat. So you see all these low carb diets these days. Well, this is the reason. It comes from this science. And just one of many, many, many things that make sugar bad is what it does to these packages that carry around fats in your bloodstream. These packages are part protein, they're part carbohydrate, they're part carbohydrate when the sugar sticks to them. They're part lipid. They are called high density lipoproteins, HDLs, and low density lipoproteins. And the cholesterol in them would be like right here. I know this is kind of small and hard to see, right? But they are like packages of cholesterol. And we've come up with the concept of good cholesterol and bad cholesterol. Cholesterol is cholesterol. It's not any different. It's not good or bad. But these packages can be more or less damaging. At least that's what we used to think. And it's still somewhat true that the low density packages also tend to stick together in your arteries and make those blood clots that can lead to a heart attack and stroke. Whereas the high density lipoproteins, the so-called good cholesterol, they actually help clean these out of your arteries. So they're actually healthy kind of fats. But now we've learned more. We've learned that these LDLs by themselves aren't so bad. It's when sugar molecules stick to them. I'll make a hexagon sugar molecule. It's when they become glycolated is the word, that they then become really sticky. And this shouldn't be hard to conceptually understand. If you have like sugar on your hands, sugary stuff, your hands get sticky after a while. Sugar is a sticky thing. It's sticky because it's got charged areas. That's what the sticking is always due to, the electrostatic force. Okay? So, anyway, this is just an example of what we've learned about these biomolecules that we're studying that can help inform our diet and so forth, and also paint sugar as the real bad guy in your diet. If you had to pick one of the worst bad guy, it would be sugar. So now, this and only right now are we going to talk about the fourth category of organic molecules called nucleic acids. I'm just going to introduce their monomer to you. It's called a nucleotide, right? What do you see here? Well, you see something that looks very carbohydrate-y. You see rings. In fact, this is a carbohydrate. But that's not all this molecule has. It has some things that are non-carbohydratish, so the whole molecule we made up another category for. Here you can see rings. But unlike the rings you saw in steroid lipids, these rings have nitrogen in the ring. And they also have some nitrogen attached. So like proteins, these have nitrogen. But unlike proteins, these have nitrogen in the rings that make them up. And then these have something that proteins don't, and that's phosphorus. But they have it attached to some oxygens in that group that we call a phosphate group, which, if you remember, phospholipids had. So if you see this, you say, okay, this might be a phospholipid or this might be a nucleotide. But then when you see this, that eliminates the phospholipid part. And actually, so does this. So this is a nucleotide, and when a bunch of these are stuck together, how do you think that's going to happen? Dehydration synthesis. This OH is coming off, another H is coming off of another one, and you'll get two of them stuck together and bunches and bunches. And this is what those molecules that we call DNA and RNA are made of. They are a nucleotide polymer. So that's all we're going to talk about them right now, just to identify them. We're going to talk about them a whole lot more later. Here's a few practice questions, okay? Enzymes are classified as which of the following? So in the syllabus specifically, they want you to know that enzymes are proteins. Just very specific, basic level 1 knowledge. We would call that definition kind of knowledge. Normally not a function of proteins. You know, I meant to put this when I had that list of protein functions. This is not. Proteins, unlike carbs and lipids, are not produced in cells to store energy. Do they have energy in them? Yes, every molecule does, because every molecule has bonds. But that's not what it was made for, and it's only if you are literally starving, I mean literally starvation starving, that you start to break down the proteins in your body for energy, and that's when your muscles start to waste away. But that's not why you made those proteins in the first place. That's just how you ended up using them. Okay, so proteins are parts of cell membranes. Those transport proteins, and proteins do transport things. So athletes concern how much protein they need, da da da. Which of the following are the basic building blocks of proteins? This is a good example of the question. There's a lot of EOC questions that are kind of like this. They have a lot of stuff up here, but you really don't need it. If you go right to the question part, you realize all they're asking me are what kind of monomers are proteins made of, and that's just, again, memorization, answer, amino acids. Now on the EOC test, we'll talk about it a lot. You don't have a time limit, so you don't worry about stuff like what I just said. But on other tests when you do, which will be just about every other test that you take, this is not a bad idea. When you have a lot of information, and that might include a graph or data or whatever, it's not a bad idea to go straight to the question to see what they're asking you. And then when you go back to read the information or the graph or whatever, you'll know what you're looking for. But in this case, you could go straight to the question and you could just answer it because you didn't need any of that information. So like complex carbs, proteins serve many functions, broken down, put back together. Both proteins and complex carbs are which of the following? Remember that the word complex means this is a polysaccharide. There are many monosaccharides put together. So are they a sequence of sugars? Yes, these are, but these aren't. So that's like many multiple choice answers. It can be partly true, but if it's not all the way true, then no good. Neither one of them are lipids. That's why we call them proteins and carbs. So that's easy to chunk out. Neither one of them are nucleotides or DNA. And so this is what we're left with that fits both of them. Okay, this is a common kind of one. They give you a list of things and ask you which combination from that list answers the question. In this case, which describes a carbohydrate, part of DNA. You should be able to X out pretty easily, which also means you can X out this one, right? They're not made of nucleotides. If you remember that nucleotide back there, nucleotides are made partly of carbohydrates, but carbohydrates are not made of nucleotides. So, these are one of the 4 types of organic molecules. So if we do this by process of elimination, which is probably often how you do these kind of things, now we're down to 2. Do carbohydrates supply energy? And this is a word I probably didn't mention before, but that complex carbohydrate, like cellulose, that we can't digest, our common name on it that you see on a food label is fiber. So they don't really supply energy in fiber. They are fiber. And then again, that one that's called cellulose is a component of plant cell walls. So both 2 and 3 fit. Oh my gosh. We don't have 2 and 3 by itself as part of one of our answers. So we better go back and look, right? We know carbohydrates are not inorganic, so we can eliminate the ones with 1. I mean, that's just a factoid. So now we got to look at these. This one doesn't have 3 in it, and we're sure that 3 is okay. So we can eliminate that for that reason. But this one has 2, 3, and 4. Are carbohydrates part of DNA? Well, you know what? Yes, they are. So I went through this one on purpose. I went through this one too fast at first, just like you might on a test, and have to go back and analyze, oh, you know what? I remember. These nucleotides, part of them is a carbohydrate, the part we call deoxyribose or ribose, OSE ending is a carbohydrate. So yes, a carbohydrate is not made of nucleotides, But DNA is made of nucleotides, and nucleotides are partly made of carbohydrates. So yes, 4 does fit. Now we got 2, 3, and 4. Disaccharides and polysaccharides have in common? Well, they're both carbohydrates. They're both made of connecting monosaccharides. Well, they're not lipids and they're not proteins, right? So that's easy, and they're not nucleic acids. So this becomes really pretty easy. They're carbohydrates, and what are they made of? Simple. Remember what that means? Monosaccharides put together by dehydration synthesis. There we go. So this is a classic kind of EOC question that they really like. They give you a big story here, right? And it's got data, it's got a picture, it's got an experiment involved, and then they ask you something about the, in this case, the experimental results. So if again you use the strategy that I talked about, go straight to the question. Based on the results, at what pH does this work best? So that's what they want to know. So if now we read the story, they talk about an enzyme, catalase. It breaks down hydrogen peroxide. You might be familiar with that, right? Breaks it down into water and oxygen gas. Now this is going to become a key because they label oxygen gas here. And if this reaction causes oxygen gas to be released, well, now we're starting to see very clearly that reaction is happening the most where there's most oxygen bubbles. Happening a little bit here and happening a little bit here, and it doesn't look like it's happening at all here or here. So can this help me answer the question, right? At these pHs, now you have to remember what acidic pHs are. They're the low ones, right? They are less than 7. Oops, I made the 7 look like the less than sign. And the basic ones are, oops, more than 7. I think I did that wrong. No mind. Okay. So now that you got that straight, at what pH does catalase work best? Well, it's this one. And then they expect you to know that neutral is pH 7, so there's your pick.